Bismillahirrahmanirrahim. Now this is another very important video on how to attempt the practical exam, which is going to be very soon on the 23rd of this month. So here are some exam skills and some practical skills in which I'm going to discuss one of the papers, which is a very recent exam. As you all know, the first important thing is that the paper is for 2 hours. So in the 2 hours that you have to do, you have got to do 2 questions. Now you might be in the paper 3.3 or the 3.4. This is of course the paper 3.5 which I'm discussing, which was for a different zone. Now the first thing and the most important thing is you read the whole question. So there's going to be a question 1 and there's going to be a question 2. So there's going to be a question 2 in which you're going to be given a slide, and this slide comes from Cambridge. Now the whole batch, whether they're 20 students, 10 students will do the question 2 first, because you see Cambridge always sends half the number of slides. So if there are 20 students in one batch, then 10 slides will only come from Cambridge. And these 10 slides, the 10 students will be selected randomly by the supervisor, which will be the teacher of the school or your bio teacher. So they will select the 10 students and then ask them to do question number 2 first. So you will do question number 2 first. Of course, you can do it in 15 minutes and then go on to question number 1. That's up to you. You can do it in 10 minutes or 15 minutes. Look at the slide and do it and start question number 1. But if you are the other group, then you will get it after 1 hour. You will get the slide after 1 hour. So this is the rule that we follow here everywhere, wherever Cambridge paper 3 is going to be conducted. Now, the most important thing is in reading the question and then treating this like a theory exam. The most important thing is that just think there is no practical, but you're doing a theory exam. So yeast cells contain the enzyme catalase, which catalyzes the breakdown of hydrogen peroxide releasing oxygen. So hydrogen peroxide in the presence of an enzyme releases oxygen. You will investigate the effect of substrate concentration on the activity of catalase in yeast. So basically the yeast is where the enzyme is present. So we are using yeast because it contains the enzyme catalase. You will need to immobilize the yeast in sodium alginate beads. When a bead containing yeast is dropped into hydrogen peroxide solution, the bead will sink. So if you have hydrogen peroxide, so this is the hydrogen peroxide. Now remember, hydrogen peroxide is the substrate here because it catalyzes the breakdown of hydrogen peroxide. So hydrogen peroxide is the substrate. So we've got hydrogen peroxide here. Now when you let that, you form a bead, and when you let the bead fall, so it says when a bead containing yeast is dropped in hydrogen peroxide, the bead will sink. As oxygen is released, the bead will rise. As oxygen is released, the bead will rise. The faster the oxygen is released, the faster the bead will rise. Now, immediately what should come to your mind is, okay, it took 10 seconds for the bead to come to the surface. Now, if it comes to the surface, remember now, it also depends upon the length of the hydrogen peroxide. If the hydrogen peroxide was only this much and you dropped the bead here, so it's only going to travel this distance. Now, keep on thinking theoretically. What are the loopholes and what are the errors in this experiment? So the length of the hydrogen peroxide is very important because it says 10 seconds, then if it comes in 5 seconds, then if it comes in 3 seconds. So now theoretically think that the time could be 10 seconds, 15 seconds, 20 seconds, 5 seconds, 3 seconds. Just think theoretically. And this will help you understand the whole question and you'll be able to do the practical very easily. You are provided with material shown in table 1.1. You must check all this as soon as you get the paper. You check all this. So Y is 7% yeast cell suspension, and this is 15 cm cube. H is 6% hydrogen peroxide solution. It's a harmful irritant, so that is a hazard. You need to wear gloves when you're handling this, or you need to wear goggles because God forbid you hit your hand with something and it spills on your face while you want to injure yourself. Then it's 2% sodium alginate solution, and then it's distilled water, and then it's 1.5% calcium chloride solution. So you see there's a percentage and there is a volume which is given to you. So you must look at the volume. You must, of course, you can ask more for the chemicals. I'm not saying that this is the only volume that you'll get. It is your right to ask for more chemicals if you need them. So don't think you're not allowed to ask for more chemicals. If any solution comes into contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection. The eye protection will probably be given to you. Carry out step 1 to step 7 to immobilize the yeast. This, of course, you must have done in the labs. I mean, I've done it with a lot with my students many times. Put 10 cm cube of C into a large test tube. What was C? Calcium chloride. So you put C, 10 cm of C in a large test tube. So this would be a test tube, not a beaker. Put 5 cm of S into a small beaker. So we have another small beaker and we have S in that. What was S? Sodium alginate. Stir Y. What was Y? Y is the 7% yeast solution. So we had that separately. Y. So you stir it because you see it starts to foam and some of it sort of settles down. So you stir Y and put 3 centimeter of Y into a beaker used in step 2. So you put 2 centimeter of that and you use that in step 2. Mix well and put 3 centimeter of Y into the beaker used in step 2. Sorry, step 2 was S. So you put Y here. Stir Y and put 3 centimeter of Y into the beaker used in step 2. Step 2 was this S beaker. So this was step 2. Use a 5 centimeter cube syringe to collect 2 centimeter of the mixture. So you use another syringe to take out how much? 2 centimeter of the mixture SY. Prepared in step 3. Now you position the 5 centimeter syringe over the large test tube containing C, shown in figure 1.1. And what do? You gently press down with the thumb to release 1 drop at a time. 5 centimeter syringe barrel resting on the top of the large test tube, mixture of S and Y, large test tube, calcium chloride solution. So this was the C. Gently press on the plunger of the 5 cm syringe with your thumb to release 1 drop into solution 3. The drop should form a bead. So immediately you will see this will form a bead. Repeat step 6 until you have used all the 2 centimeters. So we have lots and lots of beads now here. You will use these beads in step 11. You will need to carry out a serial dilution of 6% hydrogen peroxide solution, H, to reduce the concentration by half between each successive dilutions. You will need to prepare 4 concentrators of solution in addition to the 6% hydrogen peroxide, so a total of 5. And after the serial dilution is completed, you will need to have 10 centimeter of each concentration available to use. Complete figure 1.2 to show how you will prepare your serial dilution. Serial means you take it from one beaker, put it into the next beaker, then take it from this beaker. So it's called serial. That's why it's called serial dilution. Serial dilution. S-E-R-I-A-L. Serial dilution. The other is simple dilution. Figure 1.2 shows the first 2 beakers you will use to make your serial dilution. You will need to draw 3 additional beakers. For each beaker, add labeled arrows to show the volume of hydrogen peroxide transferred, the volume of distilled water added. Under each beaker, state the concentration of the hydrogen peroxide solution. So this was a very simple serial dilution wali story. So you can see here what do we have? We have 20 cm cube of 6% hydrogen peroxide. Then 10 cm of the hydrogen peroxide has been taken out and it has gone into the next one. And then we had 0 water here. but then you realize 10 has been taken out, so 10 is left behind. So we have 10%, 10 cm cube of 6% hydrogen peroxide to use. So we've got this left behind. We had 20 initially. Now we have 10 here, and 10 we have taken out, and we have put it into this test tube. Now here you will add 10 water in it. So 10 cm cube of water added. Right? Now you need to take out again 10 cm cube and add it to another beaker. And then another beaker, and then another beaker. So you'll have a total of 5. This was a 6%. This will be a 3.0% hydrogen peroxide. And here every time you would take out 10 cm cube of the 3%, and then you would add water to it, and then you would take out again from here. So you can do all the arrows first. So every time you would take out 10 cm cube, and every time you would add 10 cm cube of water into it. So this would be then 1.5%. Why? Because 3 divided by 2, because you have to half it. So this would be 1.5% H2O2. This would be 0.75. 1.5 divided by 2, because it said you have to half it. So 0.75% H2O2. And this would be 0.375. But in the last one, you'll have 20 cm cube. But in all of them, you'll have 10, because you see, you added 10 water to it, and then you removed 10 out of it. So you took 10 out of the 3%, then you took 10 out of the 1.5%, then you took 10 out of the 0.75%, and then you, of course, did not do any further. So you have now got 5 beakers, and this is called a serial dilution. So what were the 3 marks? You see, this was for 3 marks. If you look at the marks, this is for 3 marks. Now, what were the 3 marks for? Number 1, correct concentrations. Did you write 3.0, 1.5, 0.75, and 0.375? Did you write the percentages with them? Did you write the units? Where you got 1 mark for that. Then you show the transfer of 10 cm cube to each beaker, and then shows 10 cm added to each beaker. So 10 cm cube, transfer of 10 cm to each beaker, and then you added 10 cm of water to each. So that would have, of course, given you the 3 marks. Then it says carry out steps 8 to step 16. Prepare the concentration of hydrogen peroxide as described in A1 in the beakers provided. Label the small test tubes with the concentration you prepared in step 8. Put 10 centimeter of hydrogen peroxide concentration into the appropriately labeled test tube. Leave these test tubes in a test tube rack. Tip the contents of the large test tube from step 7 into a petri dish. Pick up a bead using blunt forceps. Drop the bead into the test tube containing 6%. So you took H, and how much H did you take? 10 centimeter. So you added 10 centimeter of H, it must be a certain length of the column, and you drop the beads into it. So you drop the bead into the test tube containing 6%. Start timing when the bead reaches the bottom of the test tube. So you put the stopwatch on as soon as it hits the bottom, which means this place. If the bead does not sink to the bottom of the test tube, record the time as 0. Time how long it takes for the bead to reach the surface of the hydrogen peroxide solution. So you've got to time it. How much time does it take to reach the surface of the hydrogen peroxide solution? If the bead does not reach the surface after 180 seconds, stop timing and record as more than 180. So that means you've got to only see it for 3 minutes, 180 seconds. Record the results for step 14 in A3. Repeat step 12 to step 15 with the remaining concentrations of hydrogen peroxide solution. Record your results in an appropriate table. Now I give you a simple way of doing it, is that you just do it once and then you can just cook up the results. You don't have to even do this, because there's not many marks for actually doing this practical. You can do it as a theory paper even, and you can get full marks in it. I have students who get 38 out of 40 in this paper, and this is the most highly scoring paper out of all the papers that we have. The rule is that the independent variable must be written in the first column. So you write that in the first column, and you, very important that you do not do this thing. Please remember, you do not do this thing. This is a big mistake. If you write the units in every line, that's the big mistake that you cannot make to do. So you put in all the concentrations that you've made. You've written percentage here. So you're not going to write percentage in every column. This is a -1. Do not write percentage. It says again and again in the mark scheme, no units in the body of the table. So this is the independent variable which you have put in the first column. Then the second column is the dependent variable. The dependent variable is what? What are you measuring? The time in seconds to reach the surface. So time, seconds, and no units in the body of the table. So you make this here. Now you can easily just do whatever you can. Even if you've done it, you can do the rest of it. You know what is the theory behind it. More the concentration of hydrogen peroxide, that means more the substrate, so the quicker it should rise to the surface. So the hydrogen peroxide, what we have to understand is the hydrogen peroxide, which is 6%, has more substrate in it. So the bead should quickly rise to the surface. But in 3%, if the hydrogen peroxide is 3%, that means it has less substrate in it. So this should take a little longer. So you can easily just cook up some result. Nothing very difficult. So now I have just cooked up the results, and I have just said 12 seconds, 20, 30, 42, and 80 seconds. Now the question is, what were these 5 marks for? Everybody must know what were the 5 marks for. I'm going to label them in a yellow color. First mark is for this. If you wrote percentage concentration of hydrogen peroxide, you got 1 mark, and you did not write percentage here. If you wrote percentage, you lose that mark. Then the second mark is for time in seconds. You wrote this, you got another mark. You didn't write anything else in the table. Then results for all the concentrations, you give me some results. You got another mark for that. Now time for the highest concentration, that is this one, the highest concentration was 6. So time for this should be less than this. Time for this should be less than this. Why? Because 6% hydrogen peroxide means more substrate available. So it should quickly rise to the surface. More oxygen will be produced. More enzyme substrate complex is made. More oxygen made. Oxygen, of course, makes it buoyant and it rises to the surface. It increases the buoyancy. So more the hydrogen peroxide available, more the oxygen produced, more the product. Oxygen is the product. And results recorded in whole seconds. Whole second means you write 12.0 seconds. You can't say 12.2 seconds. Please do not write that. You had to write in whole seconds. Though the stopwatch is given to you, show you a fraction as well. But please do not write the fraction. Just write 12 seconds or 14 or 20.0 or 30.0. But do not write 20.2 seconds. That is not allowed. You have to record it in whole seconds. Then part 3 says state one significant source of error in this investigation. Well, the error is, of course, it is difficult to judge when the bead reaches the top, or you can say the size of the bead varies, or you can say only one bead was used for each concentration. So these are the errors that we could have seen in this experiment. So errors are, of course, which you see is, you could have also said difficult to judge when the bead reaches the top, or the size of the bead varies, or only one bead has been used. Then it says suggest how you could make an improvement to the investigation to reduce the error which you have stated in A part 3. So you can draw a line on the test tube to show the surface more clearly, or you can use a black card, or you can measure the size of each bead and use the same size beads, or you can repeat the experiment and calculate the mean. So draw a line on the test tube to show the surface more clearly, or you can use a black card, use the same size of beads, or you repeat and calculate the mean. It depends what you wrote in the previous part of the question. You will need to estimate the concentration of hydrogen peroxide in U. You are provided with U as shown in table 1.2. U is an unknown concentration of hydrogen peroxide. Is an harmful element, 30 of it. If U comes in contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection. Carry out step 17 to step 22. Label a clean test tube U. So you label this U. Then you put 10 cm of U into the test tube. So the U that was given to you, you'll add 10 cm of that. Pick up a bead with a forceps and drop the bead into the test tube. Start timing when the bead reaches the bottom of the test tube, and then time how long it takes for the bead to reach the surface of solution U. The bead does not reach the surface, then it's 180 seconds. Stop timing and record as more than 180. Record the results from step 21 in A part 4. State the result for you. So you take any result, state the result for you. Result for you is 30 seconds. Now using your results from A2 and A5, this was sorry A5, estimate the concentration of hydrogen peroxide. Now you look at the table, where was it 30 seconds? It was this. So from 30 seconds, this was the, so you can just, whatever it is from your table, we will give you the marks for that. It isn't, there is no one answer to this. Somebody might get it 20, so then they will write 3%. Somebody might get 40, 42, so it'll say between 1.5 and 0.75. So the calculation which I have done from 30 seconds is 1.5%. You can see it from my table that it was 1.5%. So from the table, whatever your answer is, you give that and you write that, you get your 2 marks for that. 1 mark for this and 1 mark for this. Then part 7, in the procedure described in step 1 to step 16, the effect of the concentration of hydrogen peroxide on catalase was investigated. Describe how you would modify this procedure to investigate the effect of temperature on the time taken for the beads to rise, and it's 12 marks. Well, it's very simple. You use the same concentration of hydrogen peroxide. You don't change that. You use 5 different temperatures and you use a thermostatically controlled water bath. Now, everybody, please remember, whenever you're going to be talking about temperatures, you're going to write and always write using a thermostatically controlled water bath. So, in this procedure in which we're going to study the effect of temperature, we are not going to change the concentration. We'll use the same concentration of hydrogen peroxide because it said describe how you would modify this procedure, the procedure that you've just done in the previous part of the paper. So same concentration of hydrogen peroxide, you're not going to change that. You're going to use 5 different temperatures and you're going to use a thermostatically controlled. This is a marking scheme point. You must write that if at any time in the question paper there is something to do with temperature. B part of the question, immobilized enzymes are often used in industry, for example, in the production of lactose-free milk. This can increase productivity and reduce cost as the enzyme is easy to reuse and the product is not contaminated by the enzyme. A student investigated the effect of bead diameter on the hydrolysis of lactose. The beads contain the enzyme lactase. Lactase catalyzes the hydrolysis of lactose into glucose and galactose. The student put beads with a diameter of 2 mm into a syringe up to the 5 cm line. Put 5 cm milk containing lactose into the syringe. Left the syringe for 5 minutes. Measured the concentration of lactose in the milk after 5 minutes. The student used this method with a bead diameter shown in figure 1.3. So bead diameter was 2 mm, 4 mm, 6 mm, 8 mm, and 10 mm. Table 1.3 shows the results of this investigation, bead diameter and concentration of lactose after 5 minutes. Now plot a graph of the data shown on the grid in figure 1.4 and use a sharp pencil. Now the important thing is that this is the first column. This is always the independent variable, so it always goes on the x-axis. That's always the rule. The first column is the independent variable. So this is the independent variable, and whatever this is, you have to write this whole thing on the x-axis. This is the going to go on the y-axis. So you can see here I put it on the y-axis here, and you can see I have put this thing on the x-axis here. So this is, and then you plot the graph, and you will get your 4 out of 4 out of it, because there are 2 marks for the plot for the labeling of the axis, then correct plotting, and then the 5 plots joined together will get you the 4th mark. You must put the 2 millimeter like here, then 4 here, then 6 here. Every 2 centimeter it must be labeled. So it's very easy, because you see what you have to understand is that this was all that was from 2 to 10. So you've got to see that the whole grid is used. The scale will be given by Cambridge. It says scale on the x-axis 2 millimeter to 2 centimeter and labeled every 2 centimeter. Now on the y-axis you can see it started from 20.5 and it's gone up to 69%. So very easy, it was already given here 20. So then every 10 small squares or every 2 centimeter was 10. So 30, 40, 50, 60, 70, and you would fit in all the points on the graph. This scale is given to you. Cambridge is given this scale. It's in the mark schemes. The rule is that either you put a dot and a circle or you put a cross. It's either this or this. You do not confuse it. Do not circle the cross. So it's either a dot. Either you put a little dot and you put a circle around it, or you put a cross. This is the rule that we follow at Cambridge. Using the plot all the 5 points, you got another mark, and then you join the 5 points and you get another mark. You join the points with straight lines, you use a ruler, you can even use a ruler, or you can do a gentle curve through the points. I always say it's better if you just use a ruler and join point to point with the ruler so that you get your 4 out of 4 marks in the graph. It says use your graph to find the concentration of lactose in the milk after 5 minutes when the bead diameter was 5 millimeter. So you're going to do that and you find out the concentration of lactose. So you draw a line on 5 millimeter. This was 5 millimeter here. And then you draw a line and you see it, whatever it was from your graph. Explain why the percentage concentration of lactose in the milk after 5 minutes increases as the bead diameter increases. And this 3 marks is more of a theory question. So larger beads have a smaller total surface area. Larger beads have a smaller total surface area. For surface area to volume ratio, what happens to it? The smaller the bead, the greater it is. Please understand this, the surface area as the bead becomes smaller is a larger surface area. So less enzymes in contact with glucose if the larger beads have a smaller surface area, and less active sites for the substrate to bind to, lesser or fewer enzyme substrate complexes formed, decreased rate of lactose hydrolysis. So it's all theory. That completes this question 1, and I'll continue with the question 2 in the next video and wish you all the very best.